for Chapter 1, The Microbial World and You. This chapter is an introductory chapter, so most of the topics that I discuss here will be explained in greater detail later on in the semester. Now, one of the important things about science are the words, and the words carry a lot of meaning, and most of them have a basis in Latin. So if you understand the Latin root, then you get a better understanding of the word. So, going through the whole topic of this class, microbiology, if you break it down into its parts, micro means small, bio means life, and ology means to study. So basically the topic in microbiology that we study are very small living things. And these things are so small that we cannot see them with the naked eye, so we need a microscope in order to be able to see them. And one of the oldest terms for the organisms that we study in microbiology was germ. And germ means to begin, like to germinate. And now we don't use the term germ so much for these small living things. Instead, we call them microbes or microorganisms. For the first few weeks of the class, I'm going to go over the different types of microorganisms. So here, I would just like to introduce them to you briefly. The first group of organisms that I will discuss in this class are the prokaryotes. And the prokaryotes can be divided into two subgroups. The first group are the bacteria, and these you may be familiar with. These are unicellular organisms that are found all over. The second subgroup are the archaea. And the archaea are more unusual. They are found in extreme environments, so they are not as common as the bacteria. After the prokaryotes, I will be discussing various types of eukaryotic microbes. And the first group I will go through are the fungi. And with the fungi, there are two basic types, yeast and molds. And the difference between the yeast and the molds is that yeast are unicellular and molds are multicellular. So the yeast look like unicellular organisms and the molds have long strands. The next group of eukaryotic microbes that I will be discussing are the protozoa. And the word protozoa, proto means first, zoa means animal, so these are the first animals that were observed by scientists. And often we see them in pond water. So like amoebas, euglena, and paramecia. So those are the protozoa, and they can be found in water and in soil. And again, they are a type of eukaryotic microbe. Another group of eukaryotic microbes are the algae. And algae are photosynthetic organisms that are found in water. And they can be either in unicellular form or multicellular form. In the multicellular form, we usually refer to the algae as seaweed. The last group that I will be discussing are the helminths, and helminth refers to worms. And within this group, we will be discussing three types of worms, the trematodes, which are the flukes, the cestodes, which are the tapeworms, and the nematodes, which are the roundworms. And even though helminths are relatively large, you can see them with the naked eye, they do have a microscopic stage, so they are grouped together and studied under microbiology. The last group of microbes that will be discussed in class are viruses. And viruses are very, very small microbes, much smaller than a prokaryotic cell. And the unique thing about viruses is they are not considered alive, because they do not have cellular structure. And at the end of the virus chapter, we will also discuss prions. Prions are the newest microbe that has been discovered. They were discovered in the 80s. And prions are basically infectious proteins. As scientists studied living things and discovered new living things, they have tried to organize all living things into a phylogenetic tree of life. So a way to connect the living things and show how related they are. And this is one of the current phylogenetic trees of life, and it groups the living things, the larger groups that we know. Bacteria and archaea, remember, these are the prokaryotes, and then the eukarya, these would be the eukaryotes. So the bacteria make up quite a large group here. Then the archaea make up that group. And with the eukaryotes, you see the fungi, the protozoa, lots of different protozoa here. So lots of different protozoa. And the animals, and then the plants. So this is meant to show how related the organisms are. In order to organize the phylogenetic tree of life, scientists needed to be able to classify living things as to how closely they are related. So to do this, they developed a method of naming living things. And for this, they came up with the organization of kingdom, phylum, class, order, family, genus, species. So this was a way of giving names to organisms and grouping them together in a decreasing order. So for example, this is the classification, the scientific naming for humans. Humans belong to the kingdom Mammalia, the phylum Chordata, subphylum Vertebrata, class Mammalia, order Primates, family Hominidae, genus Homo, species Sapien, and so our scientific name is Homo sapien. And of course, our common name is human. So all living things are given a scientific name, and that scientific name has a genus and species part to the name. So the genus, you can think of that as the larger group name, like a last name, and the species name is a specific name. So remember, for humans, our genus name is Homo and the species name is Sapien. And for all the microbes that we discuss in class, we will be referring to them by their scientific names. Now, an important thing when you are referring to the microbes, using their scientific name, when you type them, they have to be italicized. So this is italicized. Also, another important thing is that the genus name needs to be capitalized and the species name is lowercase. And that's very important because you will be typing them on lab reports and also for your unknown project at the end of the semester. Now, the scientific names are also important because they can give you a lot of information about the microbes. So here are some examples of some scientific names of some microbes. This one we may refer to quite a bit, but Streptococcus pyogenes. And Streptococcus, the coccus refers to the shape. The shape of the cells are round, and the strepto refers to how those cells are arranged, and they're arranged in chains. And then pyogenes means it forms pus. So that's very descriptive. There are some other bacterial names that you need to know. Another one is Staphylococcus. Staphylococcus. Epidermidis. And Staphylococcus epidermidis, coccus refers to those round cells. Staphylo means irregular clusters, like kind of grapes, so that's what it looks like under the microscope. And epidermidis tells you where it lives, and it lives on the skin. So that's Staphylococcus epidermidis. Now this is a pretty long name, so when you are typing the name, the very first time you type it, you write out the whole name, Staphylococcus epidermidis, and then after that you can abbreviate it. And when you abbreviate it, it's the first letter, capitalized, of the genus name, and then you have to write out the whole last name, which is epidermidis. So you can abbreviate it that way. So anytime you are typing this name, that is the official way to type it. Sometimes, that's still kind of long, as epidermidis. Sometimes in class, I will refer to it by sort of a nickname, which is Staph Epi. but that's not an official name, and that's not how you type it on a paper. All right, so scientific names, all these microbes, we are going to refer to them by their scientific names. And again, sometimes it helps you to go through the roots to have an idea of the microbes because these names are very descriptive. Now I would like to go over a brief history of microbiology. So this is just sort of an overview of some of the major scientists and the major discoveries that were made. For this, I want you to know the scientist and their discovery or their invention, and also when they made their discovery. Was it before the Golden Age, during the Golden Age, or after the Golden Age of microbiology? This is just an overview of the history of microbiology, and you do not have to know all these dates or all these individuals, but basically microbiology started in the late 1600s, and then this is the Golden Age. So the Golden Age of microbiology was from about the mid-1800s to the early 1900s, and then of course this is after the Golden Age, and this is more recent. Hooke is the person who started the study of microbiology, and he was an English scientist and made his discovery in the mid-1600s. And remember, microbiology is the study of very small living things that we cannot see with the naked eye. So just logically, what did he have to discover to start the study of microbiology? The microscope. So he is the first person to develop a microscope to see things that you could not see with the naked eye. And the first type of organisms he observed were from plant cells, so dead organisms. And he is the first person who came up with the name cell. And cell, of course, now we know cell refers to that individual living unit. But back then, when he looked at this plant material, he thought that those divisions looked like little tiny rooms, which is what the word cell meant, like a prison cell. So that's where the word cell comes from. The next scientist, which was a little bit later than Hooke, is van Leeuwenhoek, and he was a Dutch textile merchant. And so he was very interested in using the microscopes for thread count. And what he did is he developed a more sophisticated type of microscope that he could hold up to his eye, and the light would shine through a little hole and he could look at samples through that. And in addition to advancing the microscopes and the resolution of the microscopes, he also was the first person to look at living cells. And he called those animalcules, little animals. So he was actually the first person to look at living cells. He looked at samples of pond water, he looked at samples of fecal material, and he looked at samples scraped from his own teeth. So his claim to fame is making higher resolution microscopes and actually observing the first living cells. Then much later in the 1800s, we have Jenner, and Jenner was an English physician, and his claim to fame is that he is given credit for the first vaccination. So Jenner was a doctor, and he created a vaccination protocol for smallpox. And I will be talking about him in more detail later when we talk about applications for immunology. In the 1840s, there is the Hungarian doctor Semmelweis. And Semmelweis was a doctor who worked in a maternity hospital. And at that time, when a woman went to a maternity hospital to have a baby, she had about a 50% chance of dying from childbirth fever. And at that time, of course, they didn't really understand that microbes could cause disease. So what would happen is the doctors would perform an autopsy on a woman who died of childbirth fever, trying to figure out what went wrong with her. And then when another woman went into labor, they would leave the autopsy without changing clothes, without washing their hands, and they would go assist in the birth. So of course now we know that microbes were probably being transmitted by this process. So he was the first person who came up with the idea of washing his hands in between patients. And when he did that, the mortality rate at his hospital went down by 50%. All of those scientists that I just talked about, they were laying the foundation for the golden age of microbiology. So in the golden age of microbiology, there are 3 scientists who really stand out, and that is Louis Pasteur, Joseph Lister, and Robert Koch. Pasteur is probably the most famous microbiologist, and actually he is considered the father of microbiology, and he was a French scientist in the mid-1800s. And Pasteur's contribution is that he proved biogenesis, bio-life, genesis to begin. The idea of biogenesis is that living things only come from pre-existing living things. The opposite idea of biogenesis is spontaneous generation. And spontaneous generation is the idea where you take non-living material and it can generate living things. And the classic example of spontaneous generation is leaving a piece of meat out exposed to the air, and maggots would appear on the meat. Of course, now we know that flies would lay the eggs on the meat and they would develop into maggots. But remember, back then, there weren't many microscopes, they weren't widespread, and so people could not see the eggs. So it was considered spontaneous generation. But Pasteur, he did an experiment that proved biogenesis and disproved spontaneous generation. So another example of spontaneous generation back then was when you had broth exposed to air. All of a sudden, the little animalcules, these little living cells, would spontaneously appear in the broth. So Pasteur hypothesized that the animalcules, the cells, were already present in the air. So to prove this idea, he took liquid, non-sterile liquid, and he poured it into a flask, and then he converted the top of the flask into a swan neck. So he calls those a swan-necked flask. Then once he did that, he would heat the liquid to kill all those living animalcules, and the air was forced out. And then, because of the swan neck flask, he could leave it open to the air, but any microbes that were present would be caught down here and would not be transported to the liquid. So over a long time, on the order of years, the liquid could remain sterile even though it was open to the air. And again, that's because all those microbes got trapped down there. So he proved that there was something in the air that was actually causing the broth to go bad, to spoil. And so he proved that those microbes were in the air. Lister was also important during this time of the golden age of microbiology, and he was taking ideas from Semmelweis, the hand washing, and also from Pasteur, the idea that microbes are everywhere, and he wanted to prevent infection after surgery. So his main contribution was to use antimicrobial substances on surgical wounds. So he was the first one to apply antiseptics, antimicrobial compounds to surgical wounds to reduce infection. The last person I want to discuss during the golden age of microbiology is Koch, and he was a German physician. And his contribution was to prove the germ theory of disease. Germ theory of disease. Remember, germ is the old-fashioned term for microbe, and theory of disease. And a theory, remember, a theory is something that is accepted pretty much as a fact. It's not a hypothetical. A hypothetical is a hypothesis is something that may or may not be true. A theory is accepted as true. So the germ theory of disease was the idea that these microbes can cause disease. And back then it was kind of a radical idea. Back then they felt that disease was caused mainly because God was unhappy with you, or a witch cursed you, or you breathed in some bad air. So this idea where these little animalcules, these little organisms that are found all over could actually cause disease was a very radical idea. But Koch developed a set of postulates, basically like a sequence of experimental steps that you can go through to prove that a particular microbe causes a specific disease. And so this is just an example of those steps. These are Koch's postulates, and I'll talk about them in more detail later on when we talk about disease. But it's basically investigating, identifying the microbes from diseased organisms, exposing a healthy organism to those microbes, and then that healthy organism has to come down with the same disease, the original disease, and once again you have to isolate and identify the microbe from that test organism. And Koch's postulates were very important because now scientists could go through and experimentally prove which diseases were caused by which microbes. And that is the interesting thing you see during the golden age of microbiology. So you have Koch here, Koch with the germ theory of disease, and actually he first performed this with Bacillus anthracis, the microbe that causes anthrax. But then after he did that, then you see that they're discovering all these microbes. Neisseria gonorrhoeae causes gonorrhea, Mycobacterium tuberculosis causes tuberculosis, Vibrio cholerae causes cholera, and so on. So as soon as he discovered this process, then they could start identifying the microbes that cause particular diseases. After the golden age of microbiology, there are three important scientists, Fleming, Chain, and Florey. And Fleming was a Scottish bacteriologist, and in 1928 he discovered a fungus called penicillium, which secreted a substance that killed bacteria. So he is the first person to discover penicillin, but it took about 10 years for Chain and Florey to convert that substance into a usable form so we could use it to treat bacterial infections. And 1942, in the 1940s, that's when World War II was occurring. So it was basically in World War II where we first had use of penicillin, the very first antibiotic. Watson and Crick are very, very important scientists for biology, and 1953 is a date you should know. That's when Watson and Crick made their discoveries, and you should know that they discovered the structure of DNA. So they figured out the double helix. And 1953, that was only about 70 years ago, so not very long ago. And they discovered the structure of DNA, that it's a double helix, but it took a lot longer to figure out all the processes by which DNA replicates and expresses genes, transcription and translation. But this is an important date that you should know, and Watson and Crick discovering the structure of DNA in 1953. The last scientist I want you to know is Prusiner, and in 1997 he got the Nobel Prize for discovering prions. And prions are proteins, and they cause diseases like mad cow disease. So prions were basically discovered in the 80s, and they are the newest type of microbe. And at the end of the virus chapter, that's when I will go through prions in more detail. So again, this is the overview of the history of microbiology. You only really have to know the scientists that I mentioned and their discoveries, and I want you to know if they did their research before the Golden Age, during the Golden Age, or after the Golden Age. And there are two dates that I want you to know, 1928 for the discovery of penicillin, and then 1953 for the discovery of the structure of DNA. Now I have a question for you. After hearing this basic overview into microbiology, going over the different types of microbes, the prokaryotic microbes, eukaryotic microbes, the viruses, the prions, and the history of microbiology, if you were to make a generalization, in general, are all microbes good or bad? So another way to look at it is, overall, do we benefit more from microbes or are we harmed more by microbes? So again, another way to look at it is, if we got rid of all the microbes on Earth, would it be better for us or would it be bad for us? Hopefully the answer you come up with is that overall the vast majority of microbes are good. So the vast majority of microbes actually benefit us or have no effect on us. In reality, it's very, very few microbes that are actually damaging to us or pathogenic. So what I want to do now is just go through the different benefits of microbes. The first set of benefits I want to discuss are on the ecological scale. So environmental scale, how microbes affect life on Earth. And within those ecological benefits, the first one I want to talk about is cycling of the elements. So elements are referring to the atoms that make up all living things. And living things on Earth, we do not create atoms, we don't destroy atoms. So we have to recycle the atoms that are found in living things. And probably the most common cycle you've heard of is the carbon cycle. Remember, we are organic beings made of organic molecules, and organic molecules have a high percentage of carbon. So in the carbon cycle, carbon starts out as CO2 in the atmosphere, and then that CO2 is taken up by the plants by photosynthesis, and those plants convert this carbon in the carbon dioxide into glucose, sugar, glucose, and that glucose is then eaten by the animals and used for cellular respiration. And during cellular respiration, the carbon dioxide is released back into the environment. So that is part of the carbon cycle, but the other part of the carbon cycle involves the carbon that is in the animal and plant structure. So in their bodies, in their structure. When the animals and plants die, then those structures go through decomposition. They are broken down, and that is usually done by fungi and some types of bacteria. And during this process of decomposition, the remaining carbon that is in the structure of the animals and plants, it is returned to atmospheric carbon dioxide. And so this is the cycling of the elements. So this part, the decomposition by the fungi and the bacteria, is really, really important to keep cycling carbon atoms through the carbon cycle. So every type of atom that you find in living things also goes through a similar cycle. So this is a diagram of the nitrogen cycle. And in the nitrogen cycle, you can see that you have nitrogen-fixing bacteria are very important, and denitrifying bacteria are important. Also related to this, you have algae. So algae are involved in this cycle, and also again decomposition by fungi and by bacteria. So again, to cycle nitrogen, to get nitrogen back into living things, you also need the microbes to do this too. The second ecological benefit of microbes is in terms of the flow of energy. The flow of energy refers to how energy moves through ecosystems. And you should know the basics of the flow of energy. All energy originally comes from the sun. So it originally comes from the sun, and then that light energy is converted into organic molecules by the process of photosynthesis. And then, of course, that organic energy is broken down by cellular respiration to release ATP. And once ATP is used by living cells, then it's released as heat. And heat just dissipates into the environment. That's basically entropy, so it dissipates. So flow of energy, there's one direction flow of energy. You cannot recycle energy. Now in this, you may not see any microbes, but in the photosynthetic organisms, especially the plants, it's very important to have those nitrogen-fixing bacteria. Nitrogen-fixing bacteria. And the nitrogen-fixing bacteria They provide nitrogen for the plants. The plants are unable to absorb nitrogen directly from the soil, so they need these bacteria to convert the nitrogen to a form they can absorb. So if the bacteria weren't here, the plants couldn't function, and if the plants couldn't function, we wouldn't have any photosynthesis, and if we didn't have any photosynthesis, ultimately, we wouldn't have a source of ATP. The last ecological benefit of microbes are the fact that they are important for food chains. And often they form the basis of the food chain. So how the different species in an ecosystem interact. And here's one example of a food chain in soil. And for this, you can see that there are fungi here, bacteria here, protozoa here, and then the nematodes. These are the roundworms. You have nematodes here. And so for all of this, these are all the microbes in this particular food chain, and they form a substantial part of the basis of the food chain. And this is true for all food chains, whether they are terrestrial or aquatic. The ecological benefits of the microbes in helping with the cycling of the elements, the flow of energy, and the basis for the food chain are important for life on Earth, but they're not very personalized. Your microbiota are the microbes that live on and in your body. And all of us are covered, the surface of our skin is covered in microbes, as is our digestive and respiratory systems. So in a normal, healthy person, you will have microbes all over the skin, and also in the upper digestive system and upper respiratory system and the lower digestive system. Now, for a long time, people thought that the microbes, your normal microbiota, was the cause of disease. And it is the cause of disease. Many diseases come from your own flora. So sometimes scientists thought it might be a benefit to eliminate all the microbiota because that will eliminate disease. So they performed an example on mice where they delivered some mice by cesarean section and then raised them in a microbe-free environment. And what they found is that those mice who were raised without any microbes living on their bodies, their immune system did not develop normally, they were unable to fight infection, and also their digestive system did not develop normally. So they were unable to absorb some basic nutrients. So in order for your immune system and digestive system to develop normally, you need to be exposed to microbes. You must have your microbiota. Another benefit of your microbiota, especially in your large intestines, is that you have bacteria there, like E. coli, that secretes vitamin K. So vitamin K, that's the only natural way to acquire vitamin K, and there are other microbes that also secrete vitamins. In addition to secreting vitamins, your microbes in your large intestines help you digest and absorb nutrients. The last personal benefit you have from your microbiota is something called microbial antagonism. And microbial antagonism basically means that your microbiota protects you from other pathogenic disease-causing microbes. So I've gone through the ecological benefits and then your own personal benefits of developing normally, help with digestion, vitamin production, and protecting you from pathogens. So your personal benefits. Now I want to talk about biotechnology. And biotechnology is where you use living things to create something you want. And humans have been using biotechnology on microbes for thousands of years. If you take yeast, which is a eukaryotic microbe, and give it a certain set of conditions, it will generate bread for you. And if you change those conditions, it will generate beer. There are many different types of foods that are produced using either yeast or different types of bacteria. So again, like alcohol, then you have cheese. Chocolate actually has to go through a fermentation process, different types of pickles and olives, wine, vinegar, cottage cheese, yogurt. Many of these things are produced by manipulating the microbes to perform certain reactions to generate food we want to eat. Probably initially when I said biotechnology, you were thinking more of genetic engineering. And genetic engineering is a type of biotechnology where we engineer the DNA, where we change the microbe's DNA or the organism's DNA to get a result that we want. And the very first example of this was when scientists put in a gene for a lightning bug into a tobacco plant. And they caused the tobacco plant to glow. And while that didn't have any practical applications, that proved that you can swap out genes from any species, put it into another species, and that recipient will produce the protein product. Now the way we use this is to produce most of the vaccines. We can take a gene from a virus, put it into a bacterium, and the bacterium will make the viral protein, and we can use that as a vaccine. So that is considered genetic engineering. Another benefit of microbes is bioremediation. And so bioremediation is where we use microbes to clean up pollution. And this is an example of an oil spill. So oil has spilled all over these rocks in this beach. And what these people are doing is they are spraying a bacteria that has been engineered to use the oil as food. So as they spray the microbe over the rocks, the bacteria will metabolize the oil, and when the oil is all gone, the bacteria will die off. And so microbes can be used for bioremediation. They can also be used for sewage treatment in a similar way. Another type of biotechnology or genetic engineering is to use viruses for gene therapy. And gene therapy is used to treat a genetic disease where an individual has been born with a mutation in one of their genes that causes a disease. And how gene therapy works is you would take cells from the patient, then you would use a virus to insert a copy of the healthy gene into the patient's cells. And then you take those cells and put them back into the patient, and they have been genetically engineered to have a correct copy of the gene. And there have been quite a few trials with gene therapy. They've used those for SCID, severe combined immunodeficiency, and that's where a person has a genetic disorder where they have no functional immune system. That's the boy in the bubble disease. Also muscular dystrophy, they have had clinical trials for that, and cystic fibrosis. And as far as I know, it hasn't been completely successful yet, but it has shown promise. I just want to reiterate that the vast majority of microbes are beneficial or harmless. They have no effect on us. So again, environmentally, we need the microbes to make sure life can survive on Earth. Personally, you have your normal development and normal health to thank for the microbes that live on and in your body, your microbiota. And the microbes are used in biotechnology to generate all sorts of food we want, vaccines, and possibly to cure genetic diseases. But not all microbes are beneficial. There are a few that are pathogens that harm us, they cause disease. Pathogens can be divided into two groups. The first type of pathogen is an opportunistic pathogen. And an opportunistic pathogen is one that under normal circumstances is harmless or beneficial. But if it gets to a part of your body where it shouldn't be or it overgrows, then it takes that opportunity to take nutrients from your body, to replicate in your body, and in doing so, it will cause disease. And an example of this is a staph infection. So Staphylococcus epidermidis, I talked about that bacteria before, and epidermidis tells you where it lives, and it lives on the skin. Normally, when the staph epi is on the skin, it is beneficial because it provides microbial antagonism and protects you from potential pathogenic microbes. So when the staph epi, Staphylococcus epidermidis, is on the skin, it is beneficial, helpful to you. But if you get a small cut in the skin and the staph epi can enter deeper into the epidermis, then it will cause an infection. And this is a staph infection. So Staphylococcus epidermidis is an example of an opportunistic pathogen. Another one could be E. coli. E. coli tells you where it lives. It lives in the colon, and under normal circumstances, E. coli is beneficial. It probably helped you develop normally your digestive system and immune system. Also, it provides vitamin K for you, and it performs microbial antagonism. It protects you from diarrheal diseases like C. diff. But if that E. coli migrates to the urethra, travels up to the urethra, then it will cause a UTI. So again, that's another example of an opportunistic pathogen. Most of the pathogens that cause diseases in humans are opportunistic pathogens. The other group of pathogens are called obligate pathogens. And obligate pathogens are microbes that must infect a host. So they must infect the host because that is the only way they can get nutrients or they can replicate. So they have no other option. They are obligated to infecting a host because they need to do that to get nutrients or to replicate. And often the obligate pathogens are referred to as parasites. So any of the parasitic worms are obligate pathogens. There's one group of microbes where all the individuals are obligate pathogens, and those are the viruses. Viruses do not have cellular structure. They're not considered living things, so in order to replicate themselves, they must infect a host cell, and in doing so, they're going to damage that host cell. And this is a picture of a person with HIV. And HIV is human immunodeficiency virus. So the only way for the HIV to replicate is to infect a human, and in doing so and replicating, that will cause damage to the human.